and we'll also learn the functions of SAS. And also we'll check the SAS program. I'm going to explain simple program. Okay, so how SAS work to generate the results. SAS is a programming language. How it is going to work to generate the results, we're going to learn that. And also we are going to see what is the role of this SAS in the process of clinical trial. Okay, let us see. SAS is a statistical analysis systems. It is a analysis software. Analysis as well, used for the reporting also. It is used for analysis as well for the reporting. So SAS stands for Statistical Analysis Systems. Previously it was only software, but now SAS is a company and software. So before let's say it is developed in 1960. Before 1960, there was no system, there was no computer system, there was no tool to implement the statistical processes like t-test, ANOVA. We have different statistical correlation, regression, rank, forecast. These are all the statistical processes to analyze the given data. So previously there was no tool to analyze the data. So in 1960, North Carolina University developed SAS. Initially, they developed this as for the agriculture data related issues to analyze the agriculture data. Initially, it was used in agriculture sector. But now SAS is used in different sectors. So SAS is called multi-layered. Why it is called multi-layered? It is used in multiple sectors, used in clinical, used in healthcare, used in finance, banking. Okay, healthcare, finance, banking, clinical, oil industry, gas industry, government use this one for forecasting they use. So it has many application in many sectors. Many application and many sectors. That's the reason it is called multi-layered. SAS is also called multi-database architecture. Multi-database architecture. It will connect to multiple databases. Why it has to connect to multiple databases? Why it has to connect to multiple databases? Because you will be getting the data from the client. Your client will provide you the data. When you are in SAS team, when you're working with SAS team, you will be getting the data from client. Client will give you the data, ask you to analyze. So when the client is giving you the data, your data may be in Excel, data may be in Access, data may be in Oracle, data may be in Terabase, any other, let's say data may be in CSV file. Okay, so different files you'll be getting the data. In different files you'll be getting the data. Okay, so that's the reason it is called multi-database architecture. It has to connect to multiple databases to retrieve the data, to get the data from there. To retrieve and to get the data from there. Okay, it is also called multi-vendor architecture. Multi-vendor architecture. Okay. So vendor neutral and platform independent. Vendor neutral and platform independent. That means any platform we can use SAS, any vendor can use SAS. You see, I'm working on Windows. I'm using SAS on Windows. SAS can, it will work on Linux. It will work on Unix. It will work on many other operating systems. Any platform we can use, any vendor can use the SAS. So it is called vendor neutral and platform independent. So multifunctional, SAS is called multifunctional. It has many functions, but mainly we are going to discuss 4 functions. These 4 functions will define end-to-end job of the SAS programmer. What exactly SAS programmer do? We are going to learn in this 4 functions. Okay. So first one, data access. Data access. Second one, data management. Third one, data analysis. Fourth one, data representation. Data access, data management, data analysis, data representation. What is access? As just now we discussed, when you are getting the data from the client, your client may be providing you the data in different other files. SAS has to read the data, retrieve the data from external databases, files. So what they are reading the source data, raw data. Not only reading, it will read and create the SAS dataset. SAS dataset will be into rows and columns. It's a table dataset, nothing but table. So your table will be into rows and columns. Each and every row is called observation. Each column is called variable. Okay. Each row is called observation. You can see this data. Each and every column is called variable. Each row is called observation. So this is your SAS dataset. You can easily identify it. You can see there is a square box, red color pin, red color ball like icon will be printed on the box. So this is dataset into rows and columns. So your SAS team first has to access the data into SAS environment. I have data in Excel. This is my data. So your client is providing you the data in Excel form like this, you see. The first thing that you need to do, read the data from Excel and create the SAS data set in SAS environment like this. So to analyze the data, you must have your data in this format. Okay. Directly we cannot go for analysis after accessing. We have to manage the data. Managing nothing but shape the data. Nothing but manipulating the data. Refining the data. Molding the data. Nothing but. Simple example I'll give you. See, I have source data. I have lot of variables. You see, I'm reading this data. This is side effects data. Fatigue, vomiting, abdominal pain, stroke. See, this is side effects data, adverse effects data. You know the meaning of adverse effect. So side effects, nothing but whenever you're experimenting the new drug, definitely you'll expect the side effects. So I have the side effects. But in the analysis, I don't require all these columns. I have a lot of columns, you see. See, I don't require all these columns. Out of all these, I just require only 4 to 5 columns in the analysis. Let's see. Source data consists of 1,000 rows, just example, 100 columns, 100 variables. Rows observations, columns variables. This is SAS terminology. Rows in SAS terminology called observation, column in SAS terminology called variable. Okay, so directly we cannot read. We are not going to work on all the 1,000 rows, all the 100 columns. Analysis, we don't require all these. So what do we do? We'll shape the data here. I just require only, I would like to work on only 100 observations and only 5 columns, not more than that. Only 5 columns. So this is my requirement to analyze the data. I'm going to use only this much of data in the analysis. So I have to shape the data before I come to analysis. Shaping nothing but getting the required data to perform the analysis. That is what we do in management. So refining the data to get the required data for the analysis. So this is what the data management. So we can add the new data, modify the existing data, delete the data, select the required rows and columns. If you have data in 2 tables, we merge them, we append them, we replace the data. So a lot of manipulation techniques you have in SAS. Using all these techniques, we'll get the proper data, get required data to perform the analysis. So once you get the data, you can start analyzing it. What do you mean by analysis here? So let's say when you have a raw data, we need to convert it into meaningful data, meaningful report. When you have data complex, so when you have data complex, you need to simplify the data, making it conclusive data, conclusive report, interpretative data. Let's say I have a lot of data, huge data. So we need to simplify this data and we have to make the data to the point, to the point. So this is what analysis. Analysis, we have a lot of statistical processes. Let's say ANOVA, t-test, chi-square test, frequency, means, average, summary, life test. So ranks. So these are all the statistical procedures that we implement with the SAS. Using all these, we analyze the data. When you're analyzing data, your report will be in TLF, table listing figure. Finally, you are going to generate the report in TLF, table listing figure. So let's say I have a report, but my report is in SAS format. In this format, I have a report in SAS format. You are the client, I am the programmer. So I'm sending you a report in SAS format, but you don't have the SAS software. So you cannot open that file, whichever that I'm going to send you, without having SAS software. Will you be able to open? No, right? So you need to have the SAS software to open my file. But you cannot go and buy the SAS software to open only my file, right? So as a SAS programmer, it is my job. I have to present my data. I have to make the report data easily available to end user, your client. Okay, so this is what data representation, make the data available to end user. Okay, so this is data representation. So we have to present the data in HTML, PDF, RTF. HTML, PDF, RTF. So these are all machine readable files. Means we can open these files without having any software. These are all will be opened with the simple Google Chrome. You see, I have a report in HTML. This is HTML. I can easily report like, I can easily open this. So let's see, I have a report. I can easily open here. This is PDF. So you can easily open all your reports in web link. So we don't require any software for this, even XML also. So this is what your presentation. Not only analysis, we even have to present the report in machine readable files so that end user will be able to even easily open the report, open the data. So this is what your functionality, right from accessing data, presenting the data and sending it to client. So a client will give you the data, asking you to generate the results and sending those back to them. We'll do that. We'll do by all these. So first we need to read the data, manage, analyze, present. So this is the end-to-end process. This is what the end-to-end process. Let us see a simple program. I'm going to give you one simple program and I'm going to explain how SAS program works in generating table, listing, figure. With very simple example that we are going to work with this. So I'm going to remove this one. So with the SAS functionality, we have 4 functions. Not all the people will do right from access till representation. So we have different roles in the SAS team, SAS programmer, junior SAS programmers, biostatisticians, data analyst. These are all the designations that you can apply. SDTM programmer, clinical SAS programmer, okay, SAS developer, data analyst. So let me write the program. So as you know, SAS is a programming language. As you know, SAS is programming language. So we have 2 fundamental blocks. We have 2 main blocks. So 2 main blocks, 2 fundamental blocks in the SAS programming. So which is data block, proc block. Any program that you can take, you'll have 2 blocks, data block, proc block. The data will read the data. It will retrieve the data. And what it can do, it can create a data set. Proc will process the data. Proc will do the actual analysis. and proc will produce the result. Without proc, we cannot get the result. Your output will be getting because of proc block. Proc block will generate the output. So mainly, whenever you have a raw data, finally you have to make it meaningful report. You have to make it meaningful report. So raw data will be converted to meaningful report. So initially we will work on raw data. Directly we cannot analyze. So first we'll create SAS dataset with the help of data block. Then we'll work on the SAS dataset using proc block. Okay, we'll generate the result. So raw data initially converted to SAS dataset. Then SAS dataset will be based on this. We'll work on the data and will generate the required result. So this result is meaningful report. We'll generate the report. So I'll give you one simple example. We are going to work on the treatment data. When we treat the patients with a new drug, it may cause the side effects called adverse effects. So we have the data on list of adverse effects. Your client is providing you the data and giving you the task. They just need the report on. Out of listed side effects, how many are serious side effects? How many are non-serious side effects? How many are serious? How many are non-serious side effects? So this is the requirement provided by the client. Data is given by the client and requirement also given by the client. This is the data. You see patient ID. Patient ID. Drug is given. Adverse event list. And the seriousness. This is seriousness. So patient ID 100 with the 5 mg drug. Patient is reporting fever side effect, which is non-serious. NS indicating non-serious. We can also represent it with 1 and 2. Nausea, which is non-serious. With the increasing dose 15 mg, patient showing serious side effect, stroke. In real time, we will not go on writing the data like this. In real time, we'll not write the data like this. Generally, we'll directly extract the data, import the data. Because we have a lot of data, practically, it's not possible if you go on writing like this. See, lot of side effects. So directly, we can get the data from Excel to MS Access to Oracle database to SAS. Directly, dataset will be created. So with the 20 mg, blood vomiting, B vomit, which is serious. 104 coming back to 5 mg, rash, reporting rash, which is non-serious again. Now this is the data given to you. This is the data given to you. I just need to work on this data. What is the first step I have to do? When you have a raw data, first you have to create a data set. With the help of data block, we create the data set. So this is what I'm going to do now. This is what I'm going to do. Okay. So we have, this is the window that I'm working on. It is SAS window only. SAS window has 3 main windows, you see. Editor, log, output. In detail about these windows, we'll discuss later. So initially, Editor is to write the program. Log is to check the error, warning, notes, and the syntaxes. If it is error, it will be with the dark red color. If it is warning, like this, green color. Notes will be in blue color, syntax will be in black color. No error is showing up here. Okay, so I'll show you in different window in case if you have any error. Error will be in dark red color. You can easily identify if you go to log. Okay. Output will display the result. Your result will be displayed in the output. So log will show you errors, warnings, notes. Output will display the result. Editor will help you to write the program and submit the program. So let us start with the data. Each and every statement will end with semicolon in SAS programming. As we discussed, programming language. Program is nothing but group of statements. Each statement ends with semicolon like this. Data treatment. So data statement, data block will start with the keyword data, end with RUN. End with RUN. Okay. So data set name, each and every table will be identified with the name. So we are giving the table name here, TRT. Now have to give the variable names. These are the variables, column names. To give the column names, we use input. Input, patient ID, you can see. Listing all the column names called variables. But here we have numeric variables, character variables. Numeric variable and character variable. So your numeric variable is patient ID. Character variable is drug. AE also character variable. Seriousness also character variable. So any variable after dollar represented as character. Any variable without dollar represent it is numeric variable, 0 to 9 numbers. If it is mixed like this, number and character, it will be character variable only. Now, data lines. Input will declare the variables. Data lines will declare the observations. These are the observations. Rows are called observations. No need to end observations with semicolon. After writing all these, in the next line, at a time you can use one semicolon. Okay, so this is what your data block. Data block will start with the keyword data, end with RUN. Okay, in the middle, we are using two statements. Input statement to declare the column names, data line statements to declare the observations. Okay, so let us work on proc. Before we go to proc, we'll just run the data. So select the data and there is the running man symbol you see. Okay, in running man symbol. Here you can submit, here also we can submit, you see. You can simply submit it. TRT you can see. Okay. Open TRT. TRT is created, you see. SAS dataset is created. Data block will help you to create the dataset. Dataset is created with all the variables and observations. But this is not our task, right? So our task is to generate a report, table or graph on how many are serious, how many are not serious. Let me use proc. Print. Initially, I'm just not doing any analysis. I'm not using any analysis procedure. I'll just print the data. Proc print. data equals to data set name. It will just print the data, nothing it will do. It will print the data as it is in the output. See? Printing data in output window. Okay. So, but my task is not to just print the data, right? I have to know count. Report on count of serious, count of non-serious. Frequency of serious, frequency of non-serious. In SAS, you will be getting the frequency using proc fric. Proc fric. Proc frequency will analyze the data, will give you the count of whichever the variable that you have mentioned. I'm using the statement tables, but will elaborate later. What is this table? What is this frequency and all? So simple frequency I'm giving. Proc frequency, which data set you're applying frequency analysis, and for which variable you're applying. Variable is SCR. The values that you have in SCR, you'll get the frequency for that. It will generate a table showing how many are serious, how many are non-serious, how many are serious, how many are non-serious. Okay. So now I'm going to generate the graph here. To generate the graph, we use different proc. G-chat, whichever the graph chart that you want, pi bar, h bar, b bar. I'm going to work on pi 3D. It will generate a graph showing how many serious, how many non-serious. Red portion red portion serious, blue portion non-serious. Okay, so this is what the data and proc blocks. So this is data block, will help you to create a data set. This is proc. It will process, analyze, and generate the result. Okay, so this is what your data and proc steps. So next we are going to see, I'm going to discuss this data and proc step further classes also. Initially, we are going to use the same data and proc for different examples. Today I took TRT. Tomorrow I will take EMP, employee salaries, demographic data we work on, treatment data, medication details we work on. So different data, same data, but different analysis procedure that we use. Today we use proc GChat and proc frequency. Proc means procedure. Procedure will perform the analysis, will generate the results at the same time. If you only run the data, it will not generate the result. It will just create a data set, not performing analysis. So what is the role of SAS in the process of clinical trial? For that, you need to know first clinical trial process. I'll briefly explain the clinical trial process. Whenever pharma companies, generally, who make the product? Pharma companies make the product. Whenever they make the product, they will not directly release the drug into market. They should test the drug initially on animals. Animal testing, it is called preclinical, also called non-clinical studies. So before we start clinical trial, we should get approval from FDA. This approval is called investigational approval. This approval is called investigational approval. By submitting preclinical data, we get this approval. This is the approval that we need to get to investigate the drug further on the human beings. Because further going, we are going to conduct the trial on human beings. So this is nothing but testing the drug on, testing the product on human beings. Okay, so this is to find the safety and efficacy of the drug. Establish safety and efficacy of the drug. This clinical trial will be done in 3 phases. Phase 1 will just go on increasing the number of subjects, phase 1 to 3. After completion of third phase, next step is marketing, releasing the drug into market, but will not do that directly. What we do, here also we should get approval from FDA. This approval is called marketing approval. We should submit data here, preclinical data, as well as clinical trial data. Both the data will be part of application. Okay. So even after marketing also, your drug will be monitored. There you start phase 4. Now you see the complete workflow, phase 1, preclinical to marketing. So pharma companies call sponsors. They are the one that are going to finance the clinical trial. So they ultimately get approval from the regulatory authorities. FDA in US, India it is CDSCO. Okay, we should get approval, 2 types of approval we should get. Investigational, marketing approval. Okay, so this approval will get by submitting data, but this data is raw data. This data is a huge data, and data not in a standard format, not in a format as it is requested by FDA, not in a format as per FDA, as per the regulatory authorities. Okay, it is not in a standard format. So pharma companies, what they do? Pharma companies, what they do? They need to process the data. They should process the data. Before they submit data to FDA, they should process the data first. Most of the times they will not directly process. They outsource to other company called CRO, Contract Research Organization. Contract Research Organization. Okay, CRO, Contract Research Organization. So IT companies work as CRO. Let's say pure CRO companies are there, IQVIA, Quanticate, let's say Icon. Some pharma companies also work as CRO. IT companies also work as CRO. Let's say Accenture, Tech Mahindra, HCL, okay, TCS, CTS, okay. So Novartis, Paraxel. These are all CRO only, contract research organizations. So they take up the projects from the different companies, different pharma companies. They work on the data, they submit data to FDA. Okay, so for this complete data processing, we use SAS, Statistical Analysis Systems. Okay, so what SAS do? SAS will analyze. Okay, and represent the data. They finally create TLF. Each study we require almost 150 to 180 TLFs. Okay, so whenever you get the raw data, we'll work on the data. So these standards, we follow CDISC standards. So where do we use SDTM, ADAM, where we use SDTM, ADAM, and SEND. These are all the standards that we follow when you're submitting data to FDA. SAS has to follow all these standards, prepare the data, and submit to regulatory bodies. Sometimes I'm using FDA, sometimes I'm using regulatory body. The regulatory body in US, it is FDA. India, it is CDSCO. Different countries, different regulatory bodies will be there. Okay. So when you get raw data, initially we'll work on SDTM datasets. Then we create ADAM datasets. Then from ADAM, we create TLFs. Finally submit to regulatory authorities. This is the flow. So we take one real-time project. I'm going to explain starting right from receiving raw data till submitting, till preparing TLFs. The complete process will be covered. You'll be getting raw data like this. We'll be preparing the SDTM like this. From SDTM to ADAM, then to TLFs. Okay, so this is the curriculum you see. Base and advanced will be covered. Base SAS will be covered. Advanced SAS will be covered. Clinical SAS will be covered. Base, advanced, and clinical. Okay, so in overall, it will take 3 to 4 months. This will take approximately 40 hours, 40 classes. This will take 35 to 40. We are going to work on 2 projects. Okay, so we'll work on real-time projects. Okay, 2 projects are real-time. One is pain management, other one is cancer oncology. So notes will be given, recording links will be given, version SAS 9.4 version will be given, assignment will be given topic wise, assessments will be conducted, resume preparation done, mock interviews will be done, certification guidance, dumps will be provided for the certification. Interview questions will be given. Lot of interview questions, they will be provided. Okay. So we'll work on all these. 2 projects will be covered. One I will be explaining during the sessions. Another one I'll give you at the end of the project. After completion of one project, I'll give you another project. One project I'll completely explain by writing the code and all. We'll work on TLFs, table listing figures, almost 22. So 22. So almost 22, 25 tables, listings, 7 to 8 listings, almost 10 listings. Figures we require less, let's 4 to 5 figures. Okay, so this is what the project going to cover. So any questions? So I covered the time, 3 to 4 months. What are all I'm going to cover? Okay, features of the course, curriculum of the course. In detail, curriculum will be there in website. You can have a look. Other than this, if you have any questions, you can ask, please.